Okay, so our next speaker is Marcella Williemsen. She's a PhD student at the Department of Dermatology at the University Hospital UMC Amsterdam. She was here this morning as well, and this is her second talk related to impaired interferon-gamma-induced PD-L1 expression on human vitiligo melanocytes. Please.
Hi everyone, my name is Marcella Williemsen and I'm a PhD student at the Department of Dermatology and Netherlands Institute for Pigment Disorders at the Amsterdam UMC. These coming 15 minutes, I would like to highlight a study on interferon-gamma-induced PD-L1 expression on human melanocytes that is impaired in vitiligo. Immune checkpoints, for example PD-1, modulate immune responses by regulating peripheral tolerance. Ligation of PD-L1 on antigen-presenting cells to PD-1 on cytotoxic cells leads to an inhibitory signaling, thereby preventing unwanted inflammatory responses and autoimmunity. Immunotherapy for melanoma shows that blocking PD-1 with an anti-PD-1 antibody or blocking PD-L1 with an antibody can break peripheral tolerance and reactivate an anti-tumor response in patients. Moreover, in 2 to 43% of patients with melanoma that are treated with immunotherapy, vitiligo-like depigmentation occurs, which indicates the presence of immune reactivity against both melanoma cells and melanocytes. As vitiligo can develop after treatment with anti-PD-1 or PD-L1 antibodies, this suggests the involvement of PD-1 and its ligand PD-L1 in the regulation of tolerance at the level of melanocytes. Targeting PD-1 or PD-L1 therefore seems a promising treatment strategy for human vitiligo. Impaired PD-1 or PD-L1 function is involved in a variety of autoimmune diseases, for example, type 1 diabetes, or rheumatoid arthritis, indicating the rationale to test the therapeutic potential of increasing PD-1, PD-L1 signaling in autoimmunity. In vitiligo, it is known that CD8 T cells produce high levels of interferon-gamma in perilesional skin, as is shown over here. Similarly, interferon-alpha producing dendritic cells infiltrate active vitiligo skin. Although PD-L1 expression is primarily upregulated by interferon-gamma, it can also be upregulated with type 1 interference, as shown here, in melanoma cells. Considering that interferon-gamma and type 1 interference play a crucial role in vitiligo pathogenesis, and that both are known to induce immune evasion by PD-L1 expression in melanoma, the question arises to what extent PD-L1 upregulation and resulting immune regulation occurs in vitiligo. This study therefore aims to study in situ PD-1 or PD-L1 expression in human vitiligo specimen. Secondly, we aim to investigate the role of PD-1, PD-L1 signaling in melanocyte destruction and how this is influenced by interference. To study this, we collected biopsies from patients with non-segmental vitiligo. Punch biopsies were taken from non-lesional skin, indicated here, as well as perilesional skin, which is the skin flanking the depigmented lesion. In total, we collected the material from 22 patients. 16 of these were having active disease, and these were used to study in situ PD-1, PD-L1 expression with immunohistochemistry, or they were used for a skin explant assay, which I will come to later in this presentation. The 6 stable patient materials were used to study cytokine-induced PD-L1 expression. To explore the role of PD-1, PD-L1 signaling in situ, we first immunohistochemically stained for these markers. CD3 positive cells were present in all skin biopsies, both dermal but also epidermal, as shown in this photo. Similarly, PD-1 positive cells were present in 13 out of 13 perilesional skin biopsies. And the vast majority of these PD-1 positive cells co-localized with CD3 positive cells, as shown with the black arrows, which indicate double positive cells, indicating that these are PD-1 expressing T cells. Although PD-1 positive cells were present in vitiligo skin, expression of its ligand PD-L1 was largely absent. Epidermal cells were PD-L1 negative in all specimens, whereas PD-L1 was only expressed on dermal cells in half of all the patients. And if PD-L1 positive cells were observed, even though very few cells did, PD-L1 expressing cells co-localized with CD3, so indicating PD-L1 expressing T cells. Most importantly, PD-L1 expression was not observed on epidermal melanocytes in perilesional skin biopsies, indicating that in situ, neither melanocytes nor epidermal skin cells express PD-L1, even though PD-1 was present on T cells. So since this PD-L1 expression was absent and PD-1 positive T cells were present in vitiligo skin, we investigated to what extent PD-L1 expression on human skin cells can be influenced by cytokines that are present in the skin microenvironment. As shown already before, both type 1 and type 2 interference can induce PD-L1 expression in melanoma. Also, both have clinical relevance in vitiligo pathogenesis. So therefore, we investigated both type 1 and type 2 interference. Exposure to Ifn-γ, but also type 1 interference, as shown on the right, increased PD-L1 mRNA expression on primary human melanocytes that were derived from healthy donor skin. Similarly, PD-L1 protein expression was significantly induced upon cytokine exposure. And very clear, Ifn-γ was shown to be the highest inducer of PD-L1 expression. Similar results were found for primary human fibroblasts and keratinocytes, indicating that pro-inflammatory Ifn-γ and type 1 interference can induce PD-L1 upregulation in these healthy skin cells. As Ifn-γ mRNA levels are significantly higher in depigmented lesional non-segmental vitiligo skin as compared to the pigmented non-lesional skin, and that Ifn-γ is positively correlated with PD-L1 mRNA expression in lesional vitiligo skin, we would expect PD-L1 upregulation in these patients. Exposure of skin cell suspensions from healthy donors to Ifn-γ led to an increased PD-L1 expression on both melanocytes and on melan A non-melanocytes, so other skin cells. In vitiligo, we also saw that melan A negative cells induced PD-L1 upregulation upon exposure to Ifn-γ. And also these levels were similar to the ones in healthy donors. Most interestingly, exposure to Ifn-γ did not induce PD-L1 upregulation on the melanocytes, as shown here, and also representative patients were shown in these. And here you can see that there's no upregulation of PD-L1 expression. Even though both the melanocytes, both from healthy donor and non-segmental vitiligo, had similar levels of CD119. CD119 is which encodes the Ifn-γ receptor. So therefore, it seems that vitiligo melanocytes do not upregulate PD-L1 expression upon Ifn-γ exposure, while the other cells do. As Ifn-γ producing CD8 T cells are abundantly present in lesional vitiligo skin, in situ cytokine-induced PD-L1 expression was analyzed in an autologous skin explant model. In this model, non-lesional skin biopsies were taken and cultured in either medium alone or in medium that contained CD8 positive T cells that were isolated from the perilesional skin. Skin biopsies that were cultured with medium only showed almost no PD-L1 expression. And if there was positive cells present, these were in the dermal infiltrates. In contrast, the biopsies that were co-cultured together with perilesional CD8 T cells showed a massive upregulation of PD-L1 expression, also really specifically in the epidermis, where the cytotoxic reaction takes place. Expression of PD-L1 was not restricted to the lymphocytic infiltrates only, but was seen on the majority of the epidermal cells, except for the melon-A positive cells, as indicated with these arrows. These ex vivo in situ data show that during T-cell mediated attack of melanocytes in vitiligo, no upregulation of PD-L1 occurs on melanocytes, in contrast to the other skin cells. Melanocytes from vitiligo patients thereby seem different from healthy donors in their susceptibility to melanocyte-reactive T-cell immunity. This study reveals important insights into PD-1, PD-L1 signaling in human vitiligo. We show that while PD-1 positive T cells are abundantly present in vitiligo skin, neither melanocytes nor epidermal skin cells express PD-L1. Moreover, in contrast to healthy skin cells, vitiligo melanocytes did not upregulate PD-L1 expression upon exposure to either interferon-gamma alone or in the presence of interferon-gamma producing activated T cells. Vitiligo melanocytes thereby seem capable of inhibiting attack by autoreactive T cells through PD-1, PD-L1 pathway. These data indicate that inherent differences in PD-L1 regulation in melanocytes from vitiligo patients, as compared to healthy donors, explain their selective susceptibility to melanocyte-reactive T-cell attack. With this, I would like to thank all the people involved, especially my promoter and co-promoter, Rosalie Luiten and Marcel Becking. Also, I would like to thank the patients that donated skin biopsies and the financial support from the Transscan, the Dutch Cancer Society, and the Pigment Foundation. Thanks for your attention, and if there's any questions, I'm happy to answer.
Thank you very much. This was a very interesting result that you achieved with your experiments. I have a question from the chat. These results seem interesting but counterintuitive, as pembrolizumab can induce vitiligo. Is this a compensatory response?
Yeah, I already saw the question indeed.
Yes.
I think normally there would be some autoreactive T cells maybe already against the melanocytes, but they are suppressed, for instance, by PD-1, PD-L1 signaling. And by blocking anti-PD-1, for instance, in melanoma patients, but also I think in other tumors, this can release the inhibitory effect on these T cells that will then start inducing the vitiligo in these patients. So I think normally it's a mechanism of peripheral tolerance that will keep these self-reactive T cells a bit, not active in the skin.
Thank you. I think that, yes, Thierry Passerin, Professor Thierry Passerin, you have a question?
Yeah, thank you very much. Very nice, very interesting. Any thoughts about the regulation that leads to this decreased expression of PD-L1 in vitiligo melanocytes?
No, I have actually no idea. Also from GWAS studies, it's not popping up as a gene that is affected in vitiligo, but I think that might be because it's only the melanocytes, so the melan A negative skin cells, seem to be responding to the cytokine-induced PD-L1 expression. So I think it's really a defect of the melanocytes itself, but that still is to be determined, of course. Yeah, so not really known yet, I think. Answer the questions.
Sorry, is this clear? Cherry, do you have more?
Yeah, no, that's definitely interesting. I think that maybe there's a pathway that because the expression of PD-L1 is tightly regulated, and several pathways that have been demonstrated, and I think it's more not genetics, but probably I don't know if it's a defect of melanocyte, maybe also just a deregulation by some other pathway, and that would be something that could be of interesting to target. But very nice work.
Thank you.
Thank you. We have one more question from Caroline. Caroline Lepaul.
Yeah, hi, Marcella. Just a quick question about the keratinocytes that surround the melanocytes, because if we're talking about the perilesional skin, we do think that there's quite a bit of interferon gamma present there. So why would they not be expressing the PD-L1 that could be, exhausting the T cells and help?
Yeah, I think you refer to the in situ data. There we see no PD-L1 expression at all.
Yeah.
And that's also a question we got from the reviewers. Although we took active lesions, so we would expect the inflammation still going on, I think it's a timing issue that we might have missed the high levels of interferon gamma, or just the biopsy was taken just before these were increased. So I think that might be it. Because, yeah, indeed, it's interesting that in the explant, skin explant assay, they would highly upregulate this in response to these interferon gamma producing CD8 T cells. So, I think it's a timing issue, that it's just,
Yeah, well, it would be really exciting to know more about the mechanism of the fact that they don't respond. Yeah,
Agreed.
Great, thanks.
Thank you. Any